Examples of restriction enzymes. We have example EcoR1. This EcoR1 has specific restriction site which is between G and A. So you can see that there's two strands of DNA. First, you have to recognize that this sequence is symmetrical and is palindromic sequence. Why palindromic? Because when you read it from 5 to 3, you read it as the same nucleotide sequence. For example, like 5 to 3 from one strand here, GAA, TTC, and from 5 prime to 3 prime, it is read as GAA, TTC as well. So this is palindromic sequence. And this EcoR1 has a specific restriction sites, which is between G and A. So it has to cut for both strands. So this EcoR1 cut between G and A at one strand, and another one, G and A as well, between G and A. Okay, so you're going to digest. So here they use the word digest, is same like cleave, cut, degrade, and this is the ends that they produce. This is what we call as sticky ends. Okay, when you cut it between the G and A, so this is the 5' sticky ends. Why sticky ends? Because it's not a complete cut. It is staggered. And it will produce single-stranded DNA here, single-stranded. So this single-stranded is complementary to another fragment. So it can hybridize, it can pair up, or it can complement, can undergo the complementary base pairing with other fragments. So that's why this one is single-stranded. And we have another example of restriction enzyme, which is MA1, SMA1. So this MA1, if you look at the restriction site, is between C and G. But before that, make sure that you look at the palindromic. This is palindromic sequence. When you read it from 5' to 3', it's read as CCC, GGG. And when you read it from the other strands from 5' to 3', CCC, GGG. Right, it's the same. So it's palindromic sequence and it's symmetrical. Then SMA1, the restriction site is between C and G. So it cut here between C and G at one strand. So another strand, it cuts from C, between C and G as well. So this is the ends that they produce, which is known as the blunt ends. Blunt ends is complete cut. It doesn't produce staggered. It doesn't produce a single-stranded DNA. So it's complete cut like this. This is what we call as blunt ends. Normally for restriction enzymes of cloning, we use the sticky ends. Normally the famous restriction enzymes that have been used for cloning method or genetic engineering method is EcoR1, right? So after cleavage, after digestion, some produces sticky ends like this, which is incomplete cut and staggered cut, and some produce the blunt ends, which is complete cut like this.